And it's just simple logic. You are booked for the lecture. He's also booked for the lecture. He's more senior than you. And then, but we paid. We paid. I understand we paid. All of us paid. And even them, they paid. So, emotions down, we reach a number, we go back to learning environment. Right? So, if you had emotions against me for what I said, I apologize. If I said anything wrong that hinders your feelings, I apologize. Please come and see me after the lecture. If you want me to do that, I'll do that. I've been honest. If I said anything that hinders your feelings or you felt offended, I do apologize. But what we did was abnormal.
Yes, that's addressing us regarding whatever happened. Sure.
So while you were demonstrating, I was busy trying to get us letters so that we start using this building as our official building. This is the letter coming from DOSA. So as we are doing all those things, we should remember that even us, we are not happy to be in such situations, but we are trying to bring, I really should have put that quote. Okay? So we are all trying to do the normal thing. So as we do that, let's try to be just a little bit patient. That's all we're asking. Okay, so Dane Kaukamba with the video campaign. Emotions down, like that, like I said, if you feel so offended, you want me to do that, even if you want me to do that, I'll do that. So, emotions aside, everything we said, now I want to go back to class and try and learn. That's the reason we're here. Please. So let's put our emotions aside. Whatever I was saying, forget about me for now. Now it's time to go to our class. Okay. And today we want to look at the cell. We're moving on to cell physiology. I understand and appreciate there is something that our colleagues did. Although I still think it's unnecessary, but we're going to do it. How to measure body fluid compartments. Okay, so we're going to do that in the second lecture because this one is slightly long. The next lecture is slightly shorter. So I'm going to, before I go to that short lecture, I'm going to add a few slides on measurements of body compartments. Okay. And then someone was saying, should we attend another class? Should we attend this class? It's really up to you. If you want to attend both classes, it's up to you. But I don't think it's necessary. The content, we try to normalize it as much as we can. Okay. So that's just a brief announcement. The body fluid compartments. So that's measurements. How do we measure the types of things that we use to measure? We're going to handle it in the next lecture, which is slightly shorter. This lecture is slightly longer. Okay. Do we have a mic? Is it possible to have a mic? That place is open?
Yes.
Being set up? Oh, okay. Okay, thank you. Okay. So today we want to look at cell physiology. From first year, from biology, we all know what a cell is, and we'll try to define it again today. But our major thing we're looking at now is cell physiology. in terms of our objectives, what have we come here to do? Number one, we want to kind of define what a cell is and ascertain or to acknowledge or to know that in a cell there are some things we call organelles, and those organelles have specific functions, and we might relate these functions a little bit to our clinical scenarios. Okay? I hope you're able to hear me at the back. So we will be able to relate some of these things that we're going to discuss in relation to the clinical setting. And then we also want to describe intercellular and cellular. Does that sentence sound correct? Identify intercellular and cellular extracellular connections. Okay, should have been a typo there. What we're saying is, remember when we talked about ICF and ECF, we say between cells, there is a space. That space, we called it an interstitial fluid. Now, you have cells of the skin, for example. I always give an example of the skin because that's what you give. That's what you see all the time. They are joined together. So remember, a cell is a basic unit. But this basic unit has to work together to perform its function. So when we talk about intercellular, between cells, okay, and then you have extracellular, so there are some connections or there are some organelles, or not organelles, let me say tubes, so to say, that are going to connect one cell extracellularly. So that's cell membrane, you have the junctions, they are connected. There are some organelles or structures that are going to connect intercellularly. So it's coming from the inside of this cell, going to the outside of the other side, of the other cell. So these things are going to be joined together over a long period of time. So we want to understand, how are these cells joined together? How do these cells communicate? Okay, and you have a specific lecture that's all cellular interactions and communications. Of course, we want to look at the process of exocytosis and endocytosis if times allow, and how they contribute to the normal function of the cell. This is in particular in relation to one organelle. So there's an organelle called a phagosome or a lysosome. We're going to look at those two organelles, very important for cellular function. And then these intercellular communications, so there is a way cells communicate. We want to look at that, although this is a topic for the next lecture. I don't know if we're having that lecture tomorrow or next week, whichever the class decides. So when we come to this is, I don't think we'll handle everything in today's lecture. We might just end on proteins that contribute to membrane permeability because of time. How long do I have you? Up to 12? 2 hours, okay, that's fine. Okay, so we start with basic definitions. These are things that we did a long time ago. What is a cell? By definition, we are saying that cells are the most fundamental. They are the smallest subunit of a normal body. So when we get whatever organ that we get in the body, it's made up of cells. Remember from our introduction or from our objectives, we say that these cells are interacting, they are interconnected together. For the purpose of our learning, we are picking out 2 cells. Remember that. So we are discussing a cell as one and how it's going to be connected between one cell and another. But this is happening, be it in your central nervous system, your brain, your heart, your muscle, whatever it is. Okay, so for the purposes of our discussion, we are saying that this is the smallest fundamental unit of living organisms. Okay, cells are highly specialized. What do we mean by that? In each cell, we are going to find something we call organelles. Okay, those organelles will be adapted based on the function that that cell is performing. And that's what we're saying, to say cells are highly specialized. For example, your muscle cells are working a lot. Therefore, they require what? Energy. In the cell, there is an organelle. There is something that produces energy. And therefore, muscle cells are going to have a lot of mitochondria. Why? Because muscle cells require energy. It's different from a cell such as, what cell can I give an example of? Maybe your skin. Your skin doesn't perform too much functions, therefore the number of mitochondria is going to be reduced. So we're saying that they are specialized based on the function that they are able to perform, and then they are grouped together to form tissues. So a group of cells that perform similar function. So they'll be similar in function, similar in shape, similar in the structure. When you talk about structure, we are saying what are the constituents constituents that are present in the cell. Those things are what are going to determine what tissue is going to be formed. So for example, you have skin cells. They form a tissue. They're together. They're working together. Okay. So you have different types of tissues. Brain cells. Okay. They are performing similar in function. They have a similar shape, similar organelles, and therefore they're going to form a tissue. So that's what we mean by a tissue. So when you group cells together, they're performing similar functions, then we call that a tissue. Okay. Another example there is blood, muscle. All those are examples of tissues. Now, if you have different tissues, so more than two tissues, you group them together, they form an organ. So cells, similar function and shape, you group them together, they form a tissue. You have different cells, different tissue, grouped together, still performing the same function. We're going to call them an organ. An example is the heart. Okay. The brain. So the central nervous system is made up of a lot of cells. You group them together, they form your brain tissue. Okay. So those are the types of things that we're talking about. So we can't see cells. Is our mic on now? Oh, thank you very much. I hope it's not providing echo to the other side. Hello? Okay. Yeah. So we can't see cells with our naked eyes. Like I was saying, I always give an example of the skin, because the skin is a basic thing that you see every day. So when you get your skin, you put it on a microscope. Later in your study, in anatomy, you're going to see these body cells. I think you're of course you're calling histology. Have you already studied your histology? Yes. You haven't? So when you start doing your histology is just the study of cells, microscopically. So we cannot see cells with our naked eyes. However, you and me, the old doctor, someone said the old doctor, we'll be able to see the cells in a microscope. Of course, we have two different types of microscopes, which I believe we did in first year, the electron microscope and the light microscope, right? We did that, huh? Were we able to see things in a microscope? Okay. When you come to, I think this very year, you should be able to effectively use a microscope alone, and you should be able to visualize the cells. Okay. Now, we now want to take one cell. We say that tissues are formed from same cells making the same function. Organs are made up of different tissues. We want to get one cell and discuss one cell. What is present in that cell? How does it function? What are the things that make it communicate between one cell and the other? Like I said earlier, these cells will have different organelles based on their function, but the basic structure is going to remain the same. And that's what we want to look at today. We want to look at the plasma membrane. Again, we might not be able to look at the plasma membrane today, but we'll look at it as we look at cell communication. So a cell consists of a plasma membrane. Remember, there are two types of cells. There's a eukaryotic cell and there's a prokaryotic cell. Human beings, what type of cells do we have? Eukaryotic cells. So we don't have a cell wall, isn't it? And therefore, we're going to start from a cell membrane. Now, a cell membrane, usually we call it a plasma membrane. So it's interchangeable. So don't get surprised if you have a plasma membrane or a question asking you about a plasma membrane and its properties. Okay. So we have a plasma membrane as our major organ that is surrounding the cell. We're going to look at the different functions of it. Inside that membrane, we have a fluid. When we're discussing body fluids, we call that fluid intracellular fluid, isn't it? And I think I mentioned to say what we're calling intracellular fluid is nothing but the cytoplasm. So that cytoplasm is what was having our sodium, our potassium, and all those things. And the other major factor, other major structure that is present is our nucleus. We're going to look at a bit of the function of the nucleus, which I believe most of us know, isn't it? Okay. Now, inside the cell cytoplasm, there are things we're going to call organelles. And this is where we really want to know the organelles and their functions. Okay? So organelles are basically like small structures inside the cytoplasm. Again, we can't see this with our naked eyes, but with a microscope, before the end of this year, you'll be able to see some of these things that we are discussing in your histology. So organelles are like structures. You can call them structures, you can call them a group of cells. Basically, those cells are grouped together and they perform functions, specific functions. And therefore, we need to know the function of each organelle, which we're going to look at later. Okay, so these organelles are enclosed in their own membrane. So you have the cytoplasm, so this is your cell. Now this mic is detecting the way I like teaching. This is your cell, okay? You have a cell like that. Inside your cell, you have cytoplasm, okay? Then in that cytoplasm, you have small organelles or small organs. Those organs have a cell membrane, and then inside those cell membrane, you have a group of cells performing its specific function. So very important. Organelles have a membrane that is surrounding it. Therefore, we have two types of membranes. You have the plasma or the cell membrane, and then you have the organelle membrane, which is inside each organelle. Okay. Except for the mitochondria and the nucleus, which have double membranes, individual membranes. So the mitochondria, the nucleus, those two have two membranes surrounding their structures. This is just an example or a pictorial view of the organelles that we want to discuss. On top there we have the Golgi apparatus. You don't want to preempt the lecture. Maybe this should have come at the end. Another example of organelle is the endoplasmic reticulum. We should at least know the structure and function. Okay, we have the nuclear envelope, like I was saying. The nucleus has two membranes, so it's bound by two membranes. We call that the nuclear envelope. You have the nucleolus. This is just something that is inside the nucleus. Again, we'll discuss it in subsequent lectures. Important, we have the mitochondria, which is the powerhouse. I think I was giving an example of that. Provides energy to the cell. You have the lysosomes. I think I was talking about phagocytosis, or forgetting the word I had used in my introduction. Very, very important. You have the smooth endoplasmic reticulum. You have the centrioles and the secretory granules. The centrioles form the skeleton. So the centrioles are like bone. They hold the cell together. They give it its structure. Apart from the cell membrane that we have looked at earlier. If I'm too fast, please stop me. I'm not just speaking through a mic. I'm sure my voice is pathetic in the mic. Okay, we start with a cell membrane or a plasma membrane. So I've mentioned to say the plasma membrane, the cell membrane, these two things. I don't have a picture. Okay, so that is your cell membrane. So when we say it's made up of proteins and lipids, it's a phospholipid bilayer. So it's made up of those lipids on top and the lipids below. And then it has this, you can see this bilayer. You have the lipids on top. You can see that red line there, okay? That red box on top is showing you one structure of that proteins that are formed. So now, we narrow down on that cell membrane. What is it made up of? The cell membrane. We're saying that it's made up of proteins and lipids. These proteins and lipids combine to form what we're going to bilayer. Why are we calling it a biphospholipid bilayer? We split that word. Bi means two. Okay? I wish I had a pointer. You can see the two layers there, isn't it? There's one nice layer on top there. There's another layer below there. Okay? So those lipids join together by phospholipid bilayer. One layer of proteins is on top. One layer of proteins is at the bottom. What you're seeing here is the cytoplasm. Okay? Inside here is the cytoplasm inside the cell. And then on the other side, you're going to find your ICF. Now, a very important notice here is that you can see the structure of these round phospholipids here. They're exactly the same as on the other side. So the interior and the exterior are exactly the same. The structures that are forming it. So you have that phospholipid bilayer. Okay. Did I give another example? Okay. One important thing to notice from our picture: hydrophilic heads, and then in the middle there, it's hydrophobic tails. So I take you back to your first year. Hydrophilic means what? Water loving, or they're able to allow water to pass through, isn't it? And hydrophobic is the exact opposite. So you have this inside of the cell that is water loving, or it's interacting with the ICF. And remember, ICF is just water, 80% of water. So it's literally interacting with that. It's able to allow water and other particles to pass through. And then in the middle there, you are going to have your hydrophobic tails, meaning that it's not allowing water. Or for water to pass through, it has to be allowed through the phospholipid layer. As it goes through these hydrophobic tails in the middle there, it's going to have a bit of challenge to move from one region to the other. This is very important. Why is it important? Because the cell membrane is supposed to interact with ICF. The cell membrane is supposed to interact with the ECF. However, it's not supposed to freely allow water to pass through anyhow. So this is a form of regulation. And this picture, we can see the phospholipid bilayer there. Can we see it? Yes. You can see the phospholipid heads. You can see the hydrophobic tails in the middle. This is your ICF. That is your extracellular fluid. So things like water, ion. Remember, when water is moving, it's carrying with it sodium, potassium, chloride. It's moving with the ions present. It's supposed to be regulated. And that's why you have embedded proteins inside there. You have these blue things in here. So these blue things, we're going to discuss them shortly, but these are channels through which certain ions are able to pass through. So my point being, this phospholipid bilayer, yes, it's hydrophilic at the heads, meaning that it's water-loving. It's able to allow water to pass through it freely. However, because of the hydrophilic heads that are present there, water has to, for significant amount of water to pass through the cell membrane, it has to be channeled through these nice channels that we're going to look at in subsequent slides ahead of us. Okay. Of course, we all know that it's semi-permeable, and that's why I was talking about the phospholipid bilayer. What the hydrophobic tails? If it was completely permeable, it would have been hydrophilic. So everything just comes in and passes throughout. But because of the hydrophobic tails that are binding in the middle, this membrane becomes semi-permeable. Remember, I said it's made up of lipids. Lipids are what are joining phospholipids. So phospholipids. Phosphor, that stands for phosphates. Okay, so phosphates and lipids, they join together, they form phospholipids to come and form your cell membrane. Of course, we know the major function. When someone wakes you up from your sleep, the major function of the cell membrane is to allow certain substances to pass through and inhibit certain substances to pass through. So the important terminology there is the regulation. It regulates the amount of substances or fluids that are going inside the cell and those that are coming out of the cell. Permeability varies based on the channels that are present. Okay, these are the channels I was talking about, the ones I had showed you earlier. So you have this membrane. A cell is very highly specialized. So depending on the type of cell we are dealing with, these protein channels, these channels are going to be different. In some cells, you're going to have cells with a high number of channels. In some cells, you're going to have cells with a low number of channels. Those channels that are present in the cell membrane, they are embedded in the cell membrane, they are important to regulate permeability. Depending on the number of channels that are present in the cell membrane, the permeability is going to be varied. So that's a pictorial view of what we're discussing. Okay. And that's the last bullet in there. Permeability varies because the cell membrane contains channels or regulated ion channels that are able to transport proteins, ions, and other macro or micromolecules through the cell membrane. Okay. So permeability varies based on the ion channels that are present. Please remember that. Okay. I think I've already tried to explain that it contains two layers. Those layers, we're calling them phospholipids. Why? Because they contain protein, they contain lipids. So the protein molecules are those ion channels that we're talking about. And then the lipids are like fatty structures, okay? So you're going to find fatty structures. We're going to look at what a fatty cell is, but lipids are like fat, your fat cells, huh? Okay, so you have those phospholipids. Of course, they have sugar molecules in them, sugar molecules embedded in them. The major lipids that we find are phospholipids. We don't have to know the names of those phosphatidylcholine and phosphatidylethanolamine, okay? That's just for further learning and studies, but we don't have to know those things. What we have to know is that what makes up the cell are the phospholipid bilayers. In the phospholipid bilayers, we have something we call proteins, and those proteins are going to be divided into two. Okay, we'll discuss that later. They're going to be divided into two, which are going to allow or regulate the permeability of your cell. Okay. The fourth bulletin, I think we've already discussed hydrophobic, hydrophilic. So remember, the heads, where it's interacting with the ECF, where it's interacting with the ICF is the same, and it's hydrophilic. In the middle, you have a hydrophobic layer. That is what we're talking about there. It's relatively insoluble, or it's a non-polar. Okay. What do we call a molecule that has both hydrophobic and hydrophilic properties? Amphipathic molecule, isn't it? So the possession of those hydrophilic heads, and hydrophobic tail, that hydrophobic tail, makes our cell membrane to have an amphipathic property. In terms of thickness, we don't expect you to know this, but again, you should have an idea as a doctor. What is the measurement of the cell membrane? It's about 75 nanometers. Now, for you to go to 75 nanometers, you need an electron microscope. In my life, I've never seen one. I think we only have one in Zambia, if I'm not mistaken. Yeah, an electron microscope. We should have one at UTH, only one. I've never heard another place where there's an electron microscope. So for you to know how thick a cell is, it's about 75 nanometers, and that is achieved through a microscope, isn't it? So I mentioned earlier to say, you have your hydrophilic heads, you have your hydrophilic heads at the bottom, and then you have your hydrophobic tails in the middle. In the middle there, you have some proteins that are going to be embedded in that cell membrane. Those proteins are going to be important. Remember earlier we talked about how they regulate permeability, depending on the amount of proteins that are present. Okay, so you have different proteins embedded through the cell membrane. Others are going to be on the peripheral in the ICF. Others are going to be on the peripheral in the XCF. So outside the cell membrane, they are attached to the cell membrane. Others are going to be inside the cell membrane. Others are going to cross through the cell membrane. And we call usually those ones as ion channels. So as we look at communication, we're going to look at the types of proteins that pass through the cell membrane. They are inside, they are outside. They are literally traversing the entire cell membrane. And those are important in the passage of electrolytes, so your sodium, your potassium, and the like. Based on how they are embedded in the cell membrane, you have two types of proteins. So these are normal proteins. They are all the same proteins. Of course, they are performing different functions. But this classification, yes? Please speak up.
The proteins are lacking, or water is lacking in the cell, for say, and the lack of water, the phospholipids get affected.
Yes, they do. They do. Because remember, it's water content in the cell. So yes, they do, to answer your question. Yeah. But you look at that when you look at transport across the cell membrane, and then you'll be able to discuss your question in detail. But short and long and short of it, yes, they do get affected. Okay. So you have two different types of proteins that are in the cell membrane based on where they are or based on their location. Number one, you have integral proteins. So these are proteins that are seated in this yeah, the diagram showing that. Okay, I might not have put a diagram showing that. But an example of, I don't have an example of an embedded protein, but picture a protein that is seated in between the phospholipid bilayer, okay? So such things as enzyme. Remember, the cell membrane also has cells, and those cells require enzymes, they require energy, and all those other activities that are happening in all the other cells. Remember, a cell membrane also has small molecules within it. So it requires enzymes, it requires energy, there are catalases, and all the other things that are present. So enzymes are a good example of an embedded protein. It's in between the phospholipid bilayer. Okay. Then the other group of proteins are these ones. This is a very good example. So this is an example of an ion channel. You can see this. There is a channel in between there. So it's literally communicating one cell and another cell. So you have an ion channel. What is the function of this ion channel? It's causing communication between one cell and the other. And those are what you call your integral proteins. Okay. Have I swapped the two? So the integral proteins are the ones that are embedded, and then your peripheral proteins or transmembrane proteins, these are the ones that are passing through. Very good example which we're going to look at are ion channels. They are conducting ions from the ICF to the ECF. Another example of peripheral proteins are protein pumps. Okay? Remember, we say that a cell membrane is semi-permeable. So how do proteins move from the ECF to the ICF? How do nutrients move from the ECF to the ICF? You have things like protein pumps. You have things like carrier proteins. What are carrier proteins? Glucose. So you take in your sugar, you eat your food in the morning, it gets broken down to glucose. How does that glucose move from the ICF to the ECF, where you now utilize the energy that you have? You have carrier proteins, and that's why in a condition such as diabetes times 2, it's about sugar, we've heard of people with diabetes. You have these carrier proteins that are not present, and therefore the glucose is not being transferred from your ECF, which is your blood, into your cells. So you have someone who has hyperglycemia or diabetes. Why? Because your glucose is not moving from your blood, which is your ECF, into the cells. I've talked about ion channels already. I've talked about, the other thing is receptors. Okay. So we all know that for homeostasis to be maintained, there are hormones in the body that control your cells. Those hormones are received or they are activated by receptors on the surface of the cell membrane. So those are an example of the type of proteins that we find on the peripheral or that are transversing through the cell membrane. That is just explaining what I was talking about. Is there anything we missed? We've discussed all these bulletins, isn't it? Okay, so which last one? The last bulletin, proteins is another group of functions as receptors that bind to neurotransmitters. Neurotransmitters are just your hormones, okay? Your hormones that are coming from the brain. They have an impulse. They are going to activate certain receptors on the cell. And that, how do those receptors, how do that hormone or so neuro, split that word. Neuro is a neuron, it's coming from the brain. A transmitter is just a chemical messenger, okay? So a chemical coming from the brain, we call it a neurotransmitter. How does it get received? How do cells, for example, in the ovary, get activated so that our ladies start menstruating? Okay? It's just a neurotransmitter. There is a receptor that binds that neurotransmitter, receives the message and transmits it inside the cell to the nucleus. Okay, hormones are another example. Physiological, so once these hormones or neurotransmitter bind to the cell, they initiate physiological changes. A very good physiological change is on women. Our women are supposed to menstruate. If we say we're all doctors, so there's no shy. We all know that our women menstruate, isn't it? In fact, if you're not menstruating, you should see me after this thing. But my point being, that's a physiological change. So a hormone binds to a cell and produces a physiological change. That's what we mean by physiological change. Again, we've already described this slide. We have certain proteins that are embedded inside your cell membrane. I think we call those integral proteins. Example is your enzyme there, that will catabolize. Is that the very pronunciation? Yeah, so they will catabolize different reactions, and based on the type of reaction that is taking place, you have various enzymes in between that phospholipid layer. Therefore, it shows you how complex this cell membrane is, okay? And when we start discussing the cell membrane, transport through the cell membrane, we'll be able to see some of the enzymes that cause the movement of energy, for example. That enzyme is called ATPase. Okay, I'm sure you've come across that word, isn't it? Yeah. So that's an example of an enzyme that is embedded in between that phospholipid bilayer. We've already discussed the other slides, correct? Now, at the peripheral, okay, on the membrane itself, as we are going to discuss in our lecture, we have molecules we are going to call cell adhesion molecules. Cell adhesion molecules. So I said you have this skin. This skin is made up of one cell communicating to the other cell, communicating to the other cell. How does this happen? Why is it our skin disintegrated? Because you have cell adhesion molecules, CAMs, we're going to look at that are holding these cells together. So that's another function of the cell membrane, by the way, okay? So you have these peripheral proteins that are attached to the surfaces of membranes. So you have the basement membrane, you look at that in anatomy, don't worry. A basement membrane is just something that holds different cells together, okay? An example of those peripheral proteins that attach one cell to the other or one cell to the base of something. That base, we call it a basal, should I put it? Okay, a basal membrane. Those are some of the enzymes that do that. You have the glycosated forms. Glycosated is just sugar forms of phosphatidylino, whatever, that way. You don't have to know it. But what you have to remember is you have these peripheral proteins that form cell adhesion molecules. And then some of these cell adhesion molecules are sugar-like. We all know that sugar is sticky. So when we say glycosylated, it's just sugar, glycogen, sugar. They hold together and then they form phosphatidylino-inositol. Okay, but if we split this word, okay, if we split this word in the middle, you have your phosphate there, that's your phosphate. Forgotten what the inositol is. Inositol should be a type of sugar, if I'm not mistaken. Okay, So another very important protein that is found on the surface of the cell membrane is your antigens. Okay, we all know something called white cells. We are calling them white cells in biology, right? Those cells that fight against infection. They have something called antigens on the surface. So when you, for example, when you have cholera, okay, you ingest dirty food, that food has cholera. How does your body identify that this is cholera? On the surface of your cell, you have something called antigens. So these antigens will be able to bind, or they'll be able to detect different types of antibodies that are present in your body. That's where the physiology of vaccines comes into play. Okay, so what do vaccines do? You introduce the antigen in your body, so your body becomes sensitized such that when the real antigen or when the real microbe comes, because you've already been sensitized, your body already knows that this is how this microorganism looks like. It activates its immune system quickly. Okay, cell adhesion molecules, I've already talked about that. Okay, so this is the basic structure of a cell membrane, and you should be able to define it and at least draw it, have an idea of how it looks like. Okay. We don't expect you to know that this is the type of proteins that are present in the cell, but at least we need you to remember this picture. We need you to know that it's a phospholipid bilayer, number one. The heads are hydrophilic, I'm coming. The tails are hydrophobic. In between that, you have two types of proteins. We talked about the integral proteins, those that are embedded inside your cell membrane. We talked about your peripheral proteins, okay? These are proteins that are found on the surface of the cell membrane. They can transverse through the cell membrane, or they can be on the peripheral on either side, either the ICF or the ECF. Coming to your question, I think I have two questions before we look at the functions of the cell membrane. Maybe we can entertain the question. There was a question first there. Sorry? Oh, okay. Yes?
Yes, sir. I wanted to ask, the integral proteins, are they not carrier proteins? Are they not the ones that contain carrier proteins? Because from the explanation, you're saying that the peripheral proteins are the ones that contain carrier proteins and channel proteins. So in my scenario,
okay, maybe let me just clarify that integral proteins are found inside, in between. So if you have a cell, I don't know why I didn't put a diagram here. If you have a cell membrane like this one, integral proteins are inside of the cell membrane. A good example I gave you are enzymes. Okay, enzymes. Another very good example of an enzyme is ATP. I'm sure you did ATP, correct? It provides energy. So for these carrier proteins that you're talking about at the bottom, to move those molecules, they require energy. That energy is provided by proteins that are found in between the cell membrane. The carrier proteins that we're talking about, or that we're talking about, are the peripheral proteins. Why are we calling them the peripheral proteins? Because they get something from the ECF or from the ICF and carry it to either side of the membrane. So these are not just inside the membrane, they are on either surface. Ion channels are an example of peripheral proteins. Why? Because they are found on either side of the cell membrane. They are literally transvasing, and they are allowing molecules to move from the ICF to the ECF or vice versa. Does that answer your question? Another good example were the receptors. They are found on the peripheral. In short, they are found on these phospholipid heads, okay? They are literally receiving either chemical messengers. If there is glucose in your body, they are receiving that glucose on the ECF and then transporting it through the cell membrane into the cell. There are some of the carrier proteins that just receive and deposit it inside the embedded proteins. The embedded proteins again receive that and transfer it to a peripheral protein. So it's that sequence of events that is happening. In a short form of it, peripheral proteins are found on the layer of the membrane. So it can be in the ICF, it can be in the ECF, but they can also transverse. What do we mean by that? You have a protein that is starting from the ECF all the way through the membrane and into the ICF. For the integral proteins, they are not on the peripheral. They are literally embedded in between the phospholipid bilayer. Yeah. Okay, let's finish him first.
Yes, sir. So for me, what I know is, from getting confused, I know that the integral proteins, they are found outside or inside the cell. They act like transporters, like say path for proteins and other ions to pass through them. They are not just found in between the phospholipids. That's what I think. They act as they are both found on the surface of the phospholipids.
And then the peripheral proteins, where are they found?
They are just found on the surface
Okay.
of the phospholipids.
Which reference were you using? Okay, so since it's just you and me, we're going to check the reference later, okay? Based on the reference we are using, yes? Oh, am I the one who swapped them?
I don't know, sir.
Huh?
I really struggle getting what we're trying to put across.
Okay, let me go back to this one.
Okay. It's getting interesting.
We have two types of proteins, okay? Let's get this picture very well. We have two types of proteins. We have our integral proteins, and then we have our peripheral proteins. At that point, are we together? No confusion? Okay. Then, those two types of proteins, we are saying that we are classifying them based on their location. Correct? We're saying that the integral proteins are embedded in the cell membrane. Okay? They are so our cell membrane, remember, it has two layers. We labored to talk about that. You have the phospholipid heads, and then you have the hydrophobic tails. So that is happening on this side and on the other side. That leaves you a space in between there. Is that okay? Up to that point, we're okay? Now, our integral proteins are embedded in these hydrophobic tails. They are not on the heads. In short, they are not interacting with the ICF or the ECF. They are inside this phospholipid bilayer. Those are what we are calling integral proteins. A good example of that, I didn't put an example of that.
The
I
adhesion.
put an adhesion protein. But anyway, maybe this might not be a very good example that I put. But this integral protein is in between the phospholipid bilayers. Remember, the phospholipid bilayers, one is in the ICF, one is in the ECF. On integral proteins, we have a problem there. The person who was asking the question, and there's someone who said he didn't understand. Do we understand what an integral protein is? Now, we come to something almost the same, but slightly different, based on location. We get what we're now calling a peripheral protein. Why are we calling it a peripheral protein? Remember, we've made a phospholipid bilayer. Okay? We've made a phospholipid bilayer, meaning that this is on the outside. There is a space in between there. We're calling it a phospholipid bilayer, meaning that this membrane is not just one thin paper, no. There is one portion in the middle there, that space which is containing our hydrophobic tails. That is where our integral proteins were, correct? On the peripheral, why are we calling it the peripheral? Why? Because it's on the ECF and it's on the ICF. The integral were just in between the the hydrophilic heads. And therefore, we are calling them peripheral proteins. Why? Because they are going to interact interiorly with the cytoplasm or the ICF, and then on top there, they'll be interacting with your what? Your ECF. And the examples I was giving were examples of peripheral proteins. I gave an example of receptors, okay? Why are we saying receptors? Receptors are going to be found on your ECF there at the top there. So that if a hormone comes through, I was giving an example of the menstrual cycle, it attaches to the peripheral protein, and then depending on the mechanism of which it will be transported, which is a lecture on its own in the subsequent lecture, that hormone or chemical messenger can be transported by the very protein directly into the ICF. There are some proteins that will transmit it to the integral proteins, and the integral proteins will transmit that message to the ICF. So that's what we mean by integral proteins and peripheral proteins. It's based on their location. Integral are found in between, peripheral at the bottom. And I think it's just anatomical classification, okay? Based on where they are found. Does that answer your question? Okay. We still have confusion?
No, I actually was a brief one talking about peripheral.
So that's what we mean by peripheral. We have to rush a little bit. That's what we mean by peripheral. Peripheral, we're just saying, is the ICF, is the ECF, yes? Okay, we have to kind of keep quiet so I get the question. I can't get you, please speak up.
You mentioned that antigens
Not antibodies. Not antibodies.
But then my question is, why is it that about check, why is it that when a person is getting vaccine? My question is that, for example, the COVID vaccine, they don't give it to someone who has COVID or like recently had COVID, because they feel that they make that the antibody won't.
Okay. We are going to answer that question at the end of the lecture, okay? We don't want to digress too much. We don't want to go into vaccines. I'm already behind time. Any question related to this? So just Cilia, answer your question. Huh? I'm just trying to chase time, yeah?
Yes, regarding the cell membrane, it being a double-layered, we are told to say that the phospholipids exist in a jelly-like fluid. So we want to understand what's the function of the glucose, or rather the carbohydrate molecules that are part of the phospholipid and the cholesterol.
Repeat your question, sir.
What's the function of the cholesterol and the carbohydrate molecules that are found on the cell membrane, knowing that the phospholipid exists in a jelly-like fluid? They make more like a jelly-like fluid into which molecules will float.
Did you understand the question? Yeah. Someone who understood the question. I'm not understanding this question.
I'm saying, okay, so I'm saying that the cell membrane is believed to have a phospholipid layer.
Believe or
It is. It contains to have the phospholipid layer, which is a jelly. It's a jelly because the lipid part which is there at room temperature is in form of a fluid. No wonder it's called the fluid mosaic. So I want to find out the essence of the cholesterol, it being part of the cell membrane, and the glucose molecules that are there.
His question is right there on our slides. He tells us that the cytoplasmic. Wait, I'm not going to cytoplasm. You're talking about the cell membrane.
Yes.
Okay. I don't think we're going to go into that detail to discuss the constituents of the cell membrane. It is made up of lipids. It is made up of phosphates. What you're talking about is the combination. Remember I talked about the glycated cell adhesion molecules? So this glucose is sticky, okay? So the glucose, I don't want to go back to that slide, but I think you remember the slide where I talked about glycated?
Yes.
It sticks. It's one of the ways cells adhere to one another. So this membrane is adapted. The consequence of for example, cells that have shear stress, okay, they need to be held more together. They will have a lot of glycated molecules. Why? Because they're supposed to be held more together, as opposed to a cell that is in the eye, because there is no stress there. So the composition is based or adapted based on the structure and the function of the cell. But we're not going to go into detail on why it's like that and why it's like that now. Okay, any other question? Ideally, I'm not supposed to be taking questions in the lecture, but yeah, any other question? Yeah, the answer. I'm going to pick up a few, yeah?
I'm asking about the.
Okay, guys, so we're going to take this as our 5 minutes break. So as people are asking questions, if you want to use the bathroom, please walk out and then come in. So this is like our 5 minutes break, guys, so that we kill on time. So if you want to use the bathroom, it's just that you're going to miss his question. So you can come and ask your neighbor his question. Huh? So we take this as our 5 minutes break to answer a few questions. If you want to use the bathroom, please walk out.
Oh, it's me who told them that it's not him who is supposed to do that. Those are doing
I'm asking about the transmembrane protein, which you mentioned earlier. You say the transmembrane protein spans through the cell membrane. So I was thinking that it would be the one that would act as a channel for ions to be transported through the cell. But then now you're saying it's the peripheral, which is found on the surface.
Get my explanation. I say peripheral proteins are found on the peripheral, number one. I mentioned in my explanation that they can transverse through, and I even gave an example of ion channel. I talked about sodium, I talked about potassium and chloride as examples of ions that transverse through the cell membrane. So to answer your question, yes, we classify them as peripheral proteins. Why? Because they, remember, this classification is anatomical. So we're saying that it's on the periphery. Starting from the ICF, it passes through the membrane and it's also in the ECM. However, not all proteins are transmembrane proteins. They are peripheral proteins, but they are not transmembrane proteins. Others are just on the outside, others are on the inside. Any other question? You know, this is our 5 minutes break, remember. Any other questions as we are waiting for people to settle down? Yes?
Okay. I need to feature you. Okay.
There's this question which is saying that give 5 factors that will coincide. So the cell membrane can say that you have all talked about iron.
Who can answer that question for us?
I didn't even get the question.
What factors affect cell membrane permeability? I want you to combine 2 lectures actually to answer this question. I want you to combine body fluids that we looked at previously and what we discussed today. Can someone try to answer that question? What factors influence cell membrane permeability? We're not discussing exam questions, but that's an exam question. What 5 factors affect cell membrane permeability? Let's start. Combine 2 lectures, today's lecture and the previous lecture. Someone talks about concentration. Do you agree?
temperature.
If you don't agree, then you didn't attend last time's lecture. I do agree. Who else agrees with the answer? Okay, so that's one of them. Another answer? What factors affect cell membrane permeability? Sorry? Temperature. Thank you very much. Another question? Another answer? Yes? The size of the cell, okay, yeah, of the lipid, but maybe we can say the size or the number of the embedded proteins or the peripheral proteins. Okay. Anyone else? Yes. So there are no more answers. So don't limit it to just this list. We've already given 4. I can see a lot of other hands. Okay, I think it's past 5 minutes. We can go ahead unless there's another question. We have 1 minute before we finish our 5 minutes break. Yes.
Yeah, this job of a journalist.
I hope this is the last question.
I'll get
Because our 5 minutes break is coming to an end.
Yes, I would say that the way we explain about the intact protein, the peripheral protein, somewhere behind, because the thing I know about intact proteins that they act as ion channels, and those are the examples of intact proteins, and they're embedded within the cell membrane. And the peripheral proteins, they are the one which we can find outside, the outer side of the cell membrane and the inner side of the cell membrane. And the examples, the digestive enzymes, which they are there to digest the nutrients into smaller molecules which can pass through the intact proteins.
I'll answer his question at the end of the lecture. So clear your confusion, okay? But you're going a bit away from what we're discussing, but I'll do answer your question.
Okay.
So the confusion is coming in on the integral proteins and the peripheral proteins. Maybe let me remove those words, okay? We've removed peripheral proteins and integral proteins. When we say we have proteins in the cell membrane, because people are having challenges in understanding that, I want you to realize that you have proteins that are on the peripheral of the cell membrane. We call those peripheral proteins. You have proteins that are inside the cell membrane. Between the phospholipid bilayer, you have proteins there. We are calling those the integral proteins. And we'll pause there for now. Examples of peripheral proteins and integral proteins, we'll discuss them when we look at cell transport, so that we now come to understand those enzymes that you're talking about. In the next lecture, we're going to be looking at cell transport, and we're going to be looking at all these proteins that we are discussing. So, for the purpose of this lecture, I want you to know there are 2 types of proteins. Number 1, you have your integral proteins found in between the phospholipid bilayer. And then, as the word suggests, peripheral proteins found on the side or on the surfaces of the cell membrane. The examples, I'll come and give good examples as we look at transport across the cell membrane. Those are the proteins we'll be looking at. Okay? So that we clear the air and we try to move on. Unless you have any other question. Yes. Now we have to go back to the lecture, guys. Yeah, last question.
I was hearing your explanation saying that the plasma membrane has got cell adhesion molecules. So I just need some clarity. Does that mean that including junctions?
So that is right in front of our lecture. So we're going to answer your question in just a few slides from now. Okay? Okay. So we want to now look at the functions of the cell membrane. I think it's the second phenotype of the cell membrane. One of the functions is protecting the cytoplasm and the organelles that are present. Remember, it's separating the ICF and the ECF. So there are some substances, toxic substances, drugs, for example, that will not come in as a result of your cell membrane. Remember, it is semi-permeable. So it's only going to allow certain molecules to pass through and others will not pass through. I've already talked about the semi-permeability. Barrier function, what do you mean by barrier functions? A good example is your drugs. Your drugs will only work in a certain way because of your peripheral proteins. So those who are having confusion with peripheral and integral proteins, peripheral proteins found on the surface of the cell membrane. When you take in a drug, the reason it's going to work on the heart and it's not going to work in your eyes is because on the surface of the cell membrane of the heart cells, you have receptors. And a good example of receptors are your beta receptors. So when you give a drug, it's only going to go to that peripheral protein. So it's going to attach to heart cells, because heart cells have a beta receptor, which your eyes do not have. You get it, huh? So that's the confusion you people are having with the peripheral and the integral. So that is an example of the barrier function. It prevents certain cells from receiving certain drugs, others receive certain drugs. But the best is just the barrier function. The drug is an example of a barrier function. Okay. Of course, you have your nutrients that are being transported in and out of the cell. That's straightforward. You have your gaseous exchange. Cells are respiring, okay, and they therefore need oxygen. So gaseous exchange will occur through the cell membrane. Maintenance of shape and size of the cell as well. I think that's quite straightforward. We're going to elevate our pace a little bit so that we can catch up on time. If you have questions or if I'm too fast, please stop me. Yeah? So someone was telling us about the jelly-like fluid. I don't know if it was him or him. The cytoplasm is a jelly-like fluid formed by 80% water. What concentrates the cytoplasm is body fluids. Remember when we talked about sodium, potassium, chloride, those ions will now concentrate this cytoplasm, okay? Depending on the amount of molecules that are present. But basically, you have 80% of the water. It contains a clear liquid portion, we call that a cytosol, and various particles and sizes, which we're calling organelles. And we want to discuss organelles in detail in less than an hour, okay? Other particles contain, again, we go back to body fluids, carbohydrates, proteins, lipids, electrolytes. Remember that ECF, ICF diagram we drew? So these things are what are going to concentrate your 80% water. It forms a media through which these organelles work. Remember, a cell is a basic functional unit. If one cell is not working, you get sick because a number of cells get involved. So how does that cell work? How does it produce energy? How does your nucleus produce DNA? Depends on the organelles that are present in the cell. Organelles are basically small-like structures that are going to perform different functions inside your cell. We have about 10 organelles. This list is not exhaustive. Information is changing every day, but for now, we have these 10 organelles. These ones, guys, you should know their definition and you should know their function and a bit of their structure. Okay, so we're going to discuss each one of them individually, but please go through your notes, go through your books, know them and know their structure with relation to function. Okay, so we have the nucleus, the mitochondria. We'll discuss this one by one. Now, because of time, allow me to go to the last slides because I think they're a bit more new to you people than this. Or we start with this. At least we know what the nucleus, we know what the mitochondria is. We have an example, so we don't have to struggle much. But I think things that, so I'm coming back to this. I just want to do this for the purpose of teaching. Okay, I want to start by looking at the cytoskeleton. Then I'll look at the organelles properly, one by one. Now, when we talk about the cytoskeleton, what are we talking about? For me to be standing here, I have bones that are supporting me and muscles, isn't it? And that's why I'm able to stand. For your cell to be able to maintain its shape, its structure, its function, it is supported. And that's what we're calling the cytoplasm skeleton. What holds it together? So we call that the cytoskeleton. These things hold or they maintain the structure and function of the cell. Not really function, But for the cell to function, it's supposed, for me to stand and talk in front of you like this, I'm supposed to be standing and not sleeping on the floor, correct? So for your cell to function properly, there are certain molecules, tubers, or things that hold that cell in place. We are calling these cytoplasm. So basically these are fibers, okay? They are microfibers. These ones you're able to see them with a light microscope. When you start doing your histology, you'll be able to see these fibers. So there are these small fibers that are going to hold the cell together. They're going to keep it in shape, okay? So all cells have a cytoskeleton, which is a system of fibers. So these are fibers, and we're basically discussing fibers. That not only maintains the structure of the cell, but also permits it to change its shape and move on. What do we mean by that? I'll give you an example of cells of the respiratory tract. They are continuously pushing mucus or air across them to allow you to be respiring. So they need to be moving. Another good example is your small intestine. For you to digest your food, we remember that there's that cilia from our, I don't know if it's first year or grade 12, that is continuously propelling food to move it. And therefore, it's supposed to be maintained in a certain shape and structure. These cytoskeletons also help the cell to move from one point to another, okay? So they permit it to change its shape in relation to its function, okay? And also move. A very good example of something that moves is your cilia in your respiratory tract, okay? It's continuously propelling mucus. Maybe you've not yet gone to that. Let's go to your stomach, okay? In your small intestines, I can't remember when, but you learned that there is cilia, isn't it? It propels these food molecules to allow your absorption. That's what we mean by that. There are three classes of cytoskeletal filaments, okay? Remember, we said cytoskeleton is a collection of tubes. Those tubes, those fibers are also called filaments. So there are about three types of filaments based on their diameter and the size. So you have this one cell, you have fibers that are maintaining the shape of this cell, number one. Those fibers also maintain the shape to adapt to its function. They are also important in movement of the cell. Okay, we have three types. Please let me mention here that these fibers, these microfilaments, these tubers, they are both in the ICF, in the cytoplasm. They are also on the peripheral of the cell membrane. Okay? So we have two. They have the same name, they are all fibers, but we find them both inside the cell and also on the surface of the cell. Okay? I want to clarify that so that when we look at the next slides, we avoid the confusion we created earlier. So you have tubers, microfilaments. They are both present inside the cell, in the cytoplasm. These very tubers are also present on the peripheral of the cell. A good example of that is your cilia that we were discussing earlier. So the classification of these fibers is based on the size and on the diameter. Based on those two factors, you have what you call your microfilaments, you have your intermediate filaments, and then you have your microtubers. These are basically fibers that are maintaining the structure of the cell, okay, in relation to its function. So I said the classification is based on size and diameter. So there we have the size of the microfilaments. We just need to have an idea of what they are made of. They are made of a molecule or a substance we call actin. Please, we're not going to go into detail of what actin is at this stage. We are just going to know and remember that the microfilaments are about 5 to 9 nanometers in diameter and are made up of actin. Microfilaments and microtubules, they are produced by the cell. the nucleus. Remember how proteins are produced? Okay, maybe this is why we were supposed to have started with how proteins are made by way of the ribosomes. But it's common knowledge. I think we remember from our first year we were told that proteins, you have your mRNA coming from your nucleus, goes to your ribosomes, which forms proteins. Is that too far-fetched, or remember it? We remember, so it's not something new. So your proteins, you have your mRNA coming from your nucleus, it goes to the ribosomes, and that's how proteins are made. So these microfilaments, these microtubules, are made in the same way that the other proteins are made, right? Okay, and that's what we're trying to say. So they are made like that, and then they form a cell framework. That cell framework makes our cell to adapt to the different shape and size based on the function of the cell. Intermediate, what am I explaining? Okay, I've not finished explaining that point there. Oh, actually, I was explaining another bulletin. I think it's in the next slide, sorry. This slide is trying to tell you that microfilaments and microtubules are arranged and dismantled quickly in reference to your intermediate filaments. So we have three types of filaments. You have your microtubules, you have your microtubules, microfilaments, and the intermediate filaments. Your microfilaments and your microtubules, they are assembled quickly. Yes, they are made from the nucleus, ribosomes, and they are made very quickly. So they can be assembled quickly and fast to alter the cells. Remember, we said that the cytoskeleton is important in the maintenance of a cell shape based on its function. So if a cell is supposed to change its function or it's supposed to move, these microfilaments and these microtubules are able to alternate. They're able to assemble quickly and disassemble based on the function of the cell at a particular point. So how do I try to relate that? You have your small intestines, correct? When you are eating, the small intestines have to actively move. Your cytoskeleton is actively working. When you're not eating, your cytoskeleton is more like relaxed. So these microtubules, the microfilaments, are able to alternate quickly. They're able to assemble and disassemble quickly based on the demands of the cell. In contrast to the intermediate filaments, these are more rigid. Whether the cell is actively working or is not actively working, they're kind of stuck in the same shape. The slide I was explaining, okay, I'm sure it's ahead. Microfilaments, just like microtubules, they are made up of actin filaments. These guys will be interacting with cell receptors, okay, to form focal adhesion complexes. What do we mean by that? We're going to explain what adhesion complexes are, but what we're saying is that these microfilaments, these microtubules, these tubules altogether, the cytoskeleton, are what is going to anchor the cell, first of all, to maintain its structure and function. Number two, they are going to adhere one cell to another, okay? So cells are going to be joined together. They're going to be maintained together based on the presence of these microfilaments, okay? A good example is the skin. For your skin to be held together, you have microfilaments on the surface of the cell membrane and in the cell that are holding your cell together. And that's why when you pull yourself like that, your skin still remains intact. Why? Because of the presence of the microfilaments. They are holding the cell together. Intermediate filaments, on the other hand, these we find them more at a surface we're going to call cell adhesion molecule. We're going to talk about it in the next slide. But this is where you have two cells together, they are opposed together, and they are joined together at a point where we're going to talk about gap junctions. Our colleague was talking about ion channels. So cells sometimes are able to transmit cytoplasm from one cell to another. So they are opposed together. So you have one cell and another cell joined together. And what closes this joining together? Usually you have your intermediate filaments. These allow passage of cytoplasm from one cell to another. So cells are communicating. And in some instances, cytoplasm is able to move from one cell to another. What mediates that? What forms the channel? Mainly we have something we call intermediate filaments. Of course, they are important for mechanical stress as well, but they are more predominant, they are more preserved at what we're going to call gap junctions. We'll discuss that later. In terms of diameter, they're about 8 to 14 nanometers in diameter. In their absence, cells rupture very quickly. A good example is blistering. A clinical example is something we call, I think, psoriasis. A patient with psoriasis where the skin is literally peeling off. It's as a result of this lack of cytoskeletons that are present in the cell. So you find someone comes to you and you literally see blistering. Check out the condition called psoriasis at your own time. Just check the pictures. You will see the skin and having a lot of blisters around. Okay. I don't remember this line. Okay. Okay, so basically this slide is talking about there are different types of cytoskeletons, of intermediate filaments. We can know the names based on what is made up of them and where they are found. For example, you have neurofilaments. They are found in neurons. Neurons are neuron cells, neuronal cells, cells of the central nervous system. Glio, again, these are found in your central nervous system. So this classification is more based on where you find the intermediate cells. When you find the intermediate filaments, when you find them in your nervous system or your neuronal cells, we're going to call them neurofilaments. Vermetin, you found them in the mesenchymal. You will do that in your embryology. Okay, mesenchymal is where some of your cells come from. So when you do your embryology in anatomy, you'll know what mesenchymal means. So those are endothelial cells, fibroblast, chondrocyte. These are types of cells. So this classification we're saying is based on where you find the intermediate filaments. When you find them in the cells of the CNS, neurofilaments. I don't think you've done fibroblast and the like, so I won't bother you with that. But these are things that you find in your cartilage, and in your muscle as well. Okay. Epithelial tissues such as your skin. Have we done epithelial tissue in anatomy? Yes. Yeah, so you have your desmosomes. And we'll kind of look at that just a little bit today. So this is just classifying intermediate filaments based on the cell on which they are found. Of course, we have microtubules. Again, microtubules will be important in your cell division. You remember the centromere from our first year and how it splits up the nucleus and the daughter cells? So microtubules, they all fall under the cytoskeleton. Okay. Of course, they control some movements inside the cell. Remember, we are saying that inside the cell we have different organelles. How do these organelles move? Sometimes they slide on the tubules, moving from one region to another, glucose. All those are examples of transport and distribution within the cell. But most importantly, microtubules, I want you to remember them for cell division, okay? On which the nucleus is being split up into two. This is just a pictorial view. Remember we said they are divided based on what? Diameter and size. This is the molecule, the active protein that is present. So in microfilaments, we said you have actin. In microtubules, you have tubulin. In intermediate filaments, you have a combination of both. And that's why that says several proteins. So you have actin, you have tubulin, and other types of proteins. Please remember that our cytoskeleton are just filaments or tubes that help or maintain the cell shape, and they also help it in motility. Motility can be different: chromosomal division, movement of substances across it in terms of cilia, movement of organelles and other structures inside the cell. So important: cell shape and motility. We've explained this, huh? Have we? Yes. Cilia. So basically, I was talking about a very good example of cytoskeleton is cilia, something that you are all conversant with. Very important for motility, for movement. So cilia is important for movement of either substances across the cell or the cell itself to move. It's cilia, okay, to be propelling itself. Okay. Okay, I think we've discussed this slide, unless otherwise. Maybe the only thing that we need to talk about is a bit on what cilia is made up of. Cilia is just made up of microtubules. I should have a diagram to show this. Okay, so cilia, this is your cell, that is your cilia across it. So your cilia is this one here. And this is how, when you look at it microscopically, you have two tubules that are arranged in groups of 9. And then you have another tubule at the center there. So these are what is going to eventually form your cilia. So these are just tubes. Cytoskeletons are just tubes. And this is how they look when you look at them microscopically. Forget about these diamine arms. These are just other forms of intermediate filaments that eventually make up your cilia. So cytoskeleton, a very good example of what a cytoskeleton is your cilia. And that's why I've gone into definition to talk about what that is. Flagella, these are now prokaryotic cells. Huh? So your cilia originates from as far back as your flagella. They have the same shape in terms of anatomy. They'll all be made up of microtubules and intermediate filaments. Okay. What's this slide telling us? Okay, so this is just telling us how this cilia moves. Okay. So now I don't have a pointer. You have these diene molecules, okay? These diene molecules, they are interacting together with the tubules. You don't have to remember this. This is for just, I put this for more information sake. You have the diene arms, which are just intermediate filaments. They are interacting with the tubules, which are doublet, and they are arranged in a circle. So you have a tube like that, and then you have your microtubules in a circle there, in a group of 9. And then you have 2 at the center. And this is what we mean when we say 9 + 2 arrangement. Huh? So you have a tube which is round like that, and then you have duplets of microtubules. So you have 2, 2, 2, 2, 2, 2, 9. Together they are what? 18. Then at the center you have another duplet, which is 2. That's just the structure or the way your microtubules are going to be arranged. The diene molecules are helping in terms of movement. Okay, so they coordinate between themselves and they do facilitate movement. How does that happen? You have those diene arms. Okay, you have your diene arms there. You have your microtubules. These will be interacting, probably rubbing over the other. And that movement, that motion, that interaction is what is going to foster motility. Okay. And then, still on the cytoskeleton. So we've discussed the 3 types of tubes that are found in the cytoskeleton that maintain the structure, the function, and motility. Now, these same tubules are also found on the periphery, okay? So remember your phospholipid bilayer, you have inside the cell, you have outside the cell. So when we say the peripheral, we are saying on top, those heads in the ECF, you have molecules that are a form of the cytoskeleton, meaning that you have your microtubules, you have your your microfilaments, and your intermediate filaments. These form structures on the surface of the cell membrane, and those things we're going to call them cell adhesion molecules. Why are we calling them cell adhesion molecules? Because they are going to hold two cells together. So one cell is held by another cell to form a chain. Okay, so cell adhesion molecules are just intercellular structures that are attached to the basal lamina. So you did your epithelial tissue. You have your, I'm coming to your question. You have your epithelial cells, you have the basal lamina at the bottom, isn't it? So cell adhesion molecules are attaching your epithelial cells, your skin, to your basal lamina. Remember, they are on the periphery of the cell. Apart from attaching it to the basal lamina, they are attaching it to other cells. So cells are held together as a result of what? Cell adhesion molecules. I'm saying cell adhesion molecules are intercellular structures. What intercellular structures are we talking about? Your microtubules, your intermediate whatever, and your microfilaments. They are now coming to be on the surface of the cell, and they are calling them what? In cell adhesion molecules. So they have structure and signaling. Okay, yeah. Functions. So apart from having this, so the main function of these cell adhesion molecules is to hold cells together. One cell to the neighboring cells, held together like that. However, apart from that, these cell adhesion molecules also perform, so the structure holding cells, however, some of these intercellular structures have signaling functions. Very important when we come to embryology, okay? Development of a fetus. Have you studied embryology yet?
No.
Okay, so when you come and do embryology, you find that these cell adhesion molecules become very, very important in holding tissues together. Remember, the cell is that we were produced from one cell, and that cell is growing until it forms an individual. What holds those cells together are these cell adhesion molecules. Important in inflammation. I won't go into inflammation. I'll talk about wound healing. When you hurt yourself, there are certain white things that form, holding the cells together so that you stop your bleeding, so that a new skin is formed. So those adhesion molecules are coming from those structures that we define. I've talked a lot. I hope there are no questions. Keep your questions for me behind them. Yes, there's a question. Are you sure?
Because the person has deleted it, so it's okay, I guess.
Oh, okay. Any other? Do we have any question arising from what we discussed? I hope I'm not too fast. Any questions? Okay, so we proceed. So this structure, as we've seen now in physiology, is going to be varied based on the type of cells, based on the function of those cells. So your CAMs means cell adhesion molecules are variable, okay? There are certain CAMs or cell adhesion molecules that attach to the cytoskeleton inside your cytoplasm, transverse the cell membrane, and also then attach to either the basal lamina or neighboring cells, or molecules in the intracellular fluid, extracellular fluids, and that's what we mean by that, okay? There are certain CAMs that pass through or transverse. So they are holding a structure in the cytoplasm, transverse through the cell membrane, and then hold to the basal lamina, hold to the neighboring cell, or hold to any structure in the extracellular matrix or space. All this is in a quest to maintain stability, cell adhesion. Where does the cell hold? Where does it get its basis as a result of these? These structures, please remember, are those microtubules, intermediate filaments, and The microfilaments. Okay. They bind. So the second bulletin, these microtubules can bind another microtubule outside. So they are binding the same structure. We call that homophilic binding. I'm giving an example. So it can be a microtubule, it can be a microfilament. So a microfilament attaches to a microfilament, homophilic binding, the same binding. When it attaches to other non-self structures, you have heterophilic binding. They can also attach to laminins. I think I've already explained this, where these are just proteins or a group of structures in the extracellular matrix through which these cell adhesion molecules, remember, they are providing support. So it's attached to the cell membrane and then it attaches to a family of proteins we call laminins, which are basically found in your extracellular matrix. Categories of CAMs, you have integrins, okay? These bind various receptors and are involved in the movement through interactions between integrins and the cytoskeleton. So, when we say integrins, my voice is going now. We're saying that they are heterodimers. It's a different combination. So basically, we're saying that, for example, you have microtubules, you have microfilaments. They are heterodimers. They bind to various receptors. One of the functions we talked about of cell adhesion molecules, remember, we say that in some instances, they are able to transmit signals through the cell. And that's what we mean. So those family of proteins, of cell adhesion molecules, we call them integrins. They are also involved in cell movement, but they also function as a what? As a receptor. So they have two functions. Remember, our cell adhesion molecule, the major function is stability. But in some cases, these integrins are able to conduct or are able to function as receptors. We've already talked about adhesion molecules and the IgG. IgG are just antibodies or immunoglobulins. These are important to fight infection. So they're able to detect and hold on to any microorganism that has penetrated through the body. Another example is adherens, okay? These depend on calcium 2+ for their function, okay? For them to function, they are dependent on calcium. These mediate cell-to-cell homophilic reactions. So this is like the glycocalyx, like the way sugar is bound. Huh? Sugar is sticky. It's binding two cells together. So we're saying that these adherens, they are using calcium to hold two cells together as a result of the concentration in the extracellular fluid. So you have one cell, another cell. Adherens use calcium. They concentrate calcium to cause these two cells to stick together or to hold together. And then you have selectins. These are sugars. You have your carbohydrates, and you have different types of sugars. The glycocalyx, the one that he had talked about. Okay. The class is too quiet. Am I too fast? I'm too fast. Huh? Should I slow down?
No.
No.
Have I left anyone behind?
No.
Where have I left you behind?
Ah.
I've realized I'm too fast. I might not be communicating. Am I too fast?
Ask questions, maybe.
Okay, maybe I can take some questions. Where have you left people behind? Yes. I've stopped being fast now. I'll be slow. But then I'll need 15 minutes of the time if I'm going to be slow. Yes? Yeah?
Do they move the way Cilia moves? Do they propel?
top there. So villi are just an example of cytoskeleton. They're just almost the same structure like the cilia that we talked about. They're just propelling. There's just that movement across them. Okay. Any other question? I'm told I'm too fast. I'm slowing down the lecture. Any other question? Any other question? We're going to take a break soon. Any other question?
No.
I'm too fast. I'm slowing down the lecture. Okay, so let's look at intercellular connections or junctions. Okay. Now we've described how these cells are interacting. Not really interacting, or how they are being held in stability. We are saying they are being held in stability based on the cell adhesion molecules. What makes those cell adhesion molecules are your microtubules, your microfilaments, and your intermediate filaments. Okay, so these three structures, remember, they form the cytoskeleton. And then when you have them in your peripheral, or you have them holding from cell to cell, we're now calling them what? Cell adhesion molecules. We've not changed anything. Now, the same cytoskeleton are going to form what we call intercellular connections or junctions. What are intercellular connections or junctions? These are places where cells are in contact. They are held together, so to say. Let me have a picture of this. Let me check for the picture. But from your definition, look at that. There is contact between the plasma membrane of adjacent cells. So you have one cell here and another cell here. They are being held together like that. And those things are what we're calling junctions, intercellular junctions or connections. The cytoskeleton, the very microtubules, the very intermediate filaments, and the very microtubules, they form what you call intercellular connections or junctions. The cell adhesion molecules were not holding cells tightly together, opposed like this. For these ones, they are holding the cell tightly. So you have one cell, another cell, tightly held together. That is what we're going to call an intercellular connection or an intercellular junction. Okay. Any questions from that? Yes.
Yes, sir. I wanted to ask about the glycocalyx, because you are
The
saying
glyco what?
calyx.
What's that?
It's cold. It's the sugar coat. It's a sugar coat.
Glyco? Glyco what? Okay.
A sugar coat.
It's like a fluid part of jelly, like we found on top of this. It's also like helps in connecting into their fixed structure. So I wanted to ask how they relate with the cell junctions or junctions.
Yes, there was another question?
No, it was a response to what you was asking.
Okay. Oh, thanks. There's someone who did it.
So the glycocalyx is formed.
Yes.
It's just a response.
Yeah, well, give your response.
Yes. So the glycocalyx is formed from proteoglycans and the glycosaminoglycans, those sugar molecules or derivatives that are part of the cell membrane. So their main function is to also give stability, structure, and strength. Because when you look at the content of the cell membrane, like I said earlier, the phospholipid layer, it's in form of a fluid mosaic. So things move. Okay, they're able to move from one area to the other. However, for it to have stability, strength, and structure, it needs the cholesterol and the glycocalyx, which is the carbohydrate derivatives.
Thank you. What's important is let's not go in front of the lecture. Today, we're looking at the cytoskeleton. Tomorrow, or in a subsequent lecture, we're going to look at components of the extracellular matrix. I think your question is what he had asked earlier when he talked about the glycosylated molecules, okay? And I think I tried to explain that you have things like sugar molecules that are present in your, so what he was explaining. So guys, these are what? Say that word again.
Glycocalyx.
Glycocalyx, right? So these are just fancy words. Glycogen, this is sugar, okay? So it goes back to the glycosylated. So these are sugar molecules that are holding different proteins together. So apart from this cytoskeleton, so we're saying that cytoskeleton, these are tubules that are holding cells together. In the cytoskeleton, I think we talked about calcium earlier. Was it earlier when we were talking about calcium? You also have sugar molecules. Sugar molecules sometimes form a sticky compound. And that compound is what is being referred to as what? I don't know how to pronounce that. Glycosylated. Okay? So glycosylated, you are basically saying that you have sugar molecules. Sometimes they are mixed with lipid, sometimes with fatty molecules, and they form a thicky or a sticky molecule. Okay? So that sticky molecule also provides stability to cells in between them. So that's what he was explaining. So yeah, I hope we've answered your question. Any other question on tight junctions? Please, I want you to differentiate your cytoskeleton, your cell adhesion molecules, and then I want you to differentiate your tight junctions. Those three, you need to differentiate them very well. Okay? So like I said, any other question before we move forward? Yes. So we say that the integrins have two functions. Apart from them being structural proteins, they also have receptor function. Okay. Yes. There is very minimal interstitial fluid. That's a good question, actually. So we're saying that tight junctions are holding cells closely together. They are opposing cells. So his question is, do you find any interstitial fluid? Yes, you do, but it's very minimal. And if we look at it just in a short while, we're going to look at what you're going to call gap junctions. So why these cells are held like this is to allow direct transmission of cytoplasm or ions from one cell to another without passing through the extracellular fluid. Okay? So it's like a direct form of communication between two cells. Okay. Okay. So please differentiate those three things. Huh? I'll repeat: the cytoskeleton, the cell adhesion molecules, and your tight junctions. In your tight junctions, they are closely opposed together. I'll come to your question. They are divided into two. Okay, so these tight junctions are divided into two. The first broad category, these are molecules that hold cells together. They literally oppose them together, and this is very important. We'll look at this in a different, in the next slide. So for example, you have receptors on the top, which are not supposed to be at the bottom. You have certain proteins that are supposed to be held on the periphery, on the ECF, which are not supposed to move inside to become integral proteins or to move in the ECF. So these tight junctions, the first one, the group, we're calling, okay, we didn't call them the name. Okay, the first group that holds structures together. So it's going to hold cells together for the very importance of not allowing proteins. This is just an example in case someone misquotes me. So proteins, for example, that are supposed to be on the apical surface, the tight junctions help hold these proteins and maintain them on the surface. Okay. A good example of those are these four, although practically in our lesson we'll look at the three. You have the tight junctions, we call them the zonal occludens. You have the desmosomes, we call them and the other one is the hemidesmosomes. Desmosomes, hemidesmosomes almost have a similar function and also in relation to structure. And then you also have your focal adhesion molecules. So we're now going to expand these, okay? Then The other category is what I was trying to explain earlier on. These ones, remember, the ones that are holding cells together, there are other ones that are going to hold cells together, but apart from holding cells together, they are going to form a channel between two cells, okay? So you have like, these are your microtubules, those particularly gap junctions. So you have gap junctions where you have cytoplasm diffusing. We look at the first broad of categories. Remember we said we have four. We have tight junctions, we have the desmosomes and the heavy desmosomes. Okay? Now, these are found at the apical surface. Let me give you a picture. Okay? So you can see that tight junctions at the apical surface. I was giving an example of how they hold certain proteins together. For your cilia to be maintained at the top there. Imagine cilia is at the bottom or it's in the intracellular fluid. Then we have chaos, isn't it? Your cell is not going to work. So you have your tight junctions that are going to hold. You can see them, they are holding, maintaining certain proteins that are supposed to be at the apical surface to be there. We talked about two types of proteins in the cell. We talked about the apical proteins and the integral proteins. The apical proteins are maintained at the surface or at the top by tight junctions. Of course, it will not allow certain cells to pass through. So these tight junctions can also be between two cells. So you have a cell here, you have another cell here. So you have a cell here, you have another cell here. Imagine this is shorter, very minimal. So if you have your two cells here, these sometimes will prevent, of course they permit, they prevent movement of proteins in the plane of the membrane. So you don't want certain proteins to move from the apical surface to the bottom. You want them to be maintained at the apical surface. And what holds those surfaces are your tight junctions. There are certain proteins that you also just want to be at the bottom. And therefore, these tight junctions will keep those proteins at the basolateral surface, bottom surface of the epithelium. Okay. Of course, you have three main types. You have your occludins, your junction or adhesion molecules, and your claudins. We don't expect you to know a lot of details of what is making up the claudins and everything, but at least you should have an idea of what we're talking about. And then you have your zonular adherens and your desmosomes. Okay. Okay, do I have the picture? Let me go back to the picture. You can see the zonular adherens there. They are kind of a form of or a continuation of the tight junctions. Okay. So at the apical surface or the basal surface, you have your tight junctions. As you approach in the peripheral, as you come down there, it's kind of a continuation of your tight junctions. So you're coming down there, and then you're going to find your tight junctions. These are, remember, the tight junctions are keeping the apical proteins on top. In the, what plane is this? This is a vertical plane. So your adherents are kind of preventing protein movement or movement of molecules in the horizontal plane. Okay, and that's what we mean by that. They maintain transporters and channels. Am I going back to apical? So on the basal side, what I mean is this part. This like on the adjacent cells. So you have your tight junctions at the bottom on the basal side, on the lateral side here. You have the continuation of what? Your zonular adherens. Your desmosomes are joining two cells together on the lateral side as well. Okay? Two adjacent cell membranes. So you have your cell here, you have your cell. What is holding these two cells together is what? Your desmosomes or your macular adherens. Now, you have between two membranes thickening, the intercellular space contains cadherins and other transmembrane proteins. In fact, this was the slide I was trying to share with him. The question he was asking. Between two cells, apart from these other molecules, you have proteins, transmembrane proteins, such as the one he mentioned, the glycocalyx, whatever. Those things are just sugar molecules that hold two cells together. Okay. It's almost 12 hours and we've not even done half the lecture. And then finally, the other type of communication are the gap junctions. These ones you should know and remember. Why am I saying that these ones you should know and remember? Is because these are allowing molecules to pass through cells. So it's a form of communication. So cells are communicating between neighboring cells without them having to involve the extracellular fluid. So you have one cell here, one cell here. Cytoplasm is moving from one cell to another cell without passing through the extracellular fluid. This is very important for rapid propagation of electrical activity, especially when we come and look at the nerve cell or the nerve tissue. Neurons are supposed to be transmitted very, very rapidly. So for me to lift my hand like this, remember, a chemical impulse has to move from my brain to my hand to tell it to rise like that. So to make this connection or this communication very faster, you have something you call gap junctions where cells interact very directly without having to move through to the ECF. Any questions? I have 10 minutes to 12 hours. Any questions so far? So this is an example of a gap junction. You have two cells. How is this diagram? Okay, so you have one cell on this side. You have one cell at the top there. And then cytoplasm is able to move in either direction. So it can come from out there to the other one or from here to the other side. So they are literally transversing across each other. Cells are communicating without them having to pass through the cytoplasm. In terms of the anatomy, what is it made up of? It's made up of structures you call connexons, which are hydrophilic channels. Hydrophilic, basically water-loving, and we know that your cytoplasm is hydrophilic, isn't it? So they are basically connecting two cells together. It's made up of 6 subunits. Again, we go back to our microtubules, our intermediate filaments, and our microfilament. So those channels hold the cell together and allow communication between them. Okay. That ends the lecture without the slides we missed. I thought it was very important to discuss the cytoskeleton because I didn't want to rush it through. Now I need 15 minutes, just 15 minutes to discuss cell organelles and we're done with cell physiology. Okay, is that fine? Should we take a break?
Yes. Oh no, sir.
Okay, we can take a break in forms of questions. So just 3 minutes break. Then we go back to discuss the organelles. Okay. Any questions? Okay, guys, this at least you already know, so it will be very fast. We all know what an endoplasmic reticulum is. We all know what a nucleus is. So those are not new things. Under 15 minutes to be done. Yes. So we're taking a 5 minutes break, and then I'm just going to be here for 15 minutes, and then we dismiss the class.
My question is that I first had a question.
Please, go ahead.
I wasn't able to understand how you're relating the cell adhesion molecules with the cytoskeleton. Is the cytoskeleton the cell adhesion molecules, or the cell adhesion molecules are connected to the cytoskeleton? And then my other question is that when it comes to the cytoskeleton, is it that it's found inside or it's just found like in certain areas, like let's say this, when the cell has got cilia outside, then we're going to have the cytoskeleton in there. Or yes, and is it that a cell is going to involve all the types, like we're going to have microfilaments in there, the intermediate filaments and the microtubules, or we're just going to have a certain type depending on the type of cell?
Maybe we take two questions so that we answer them at the same time. We're remaining with 2 minutes before our break finishes.
Question is on the gap junction. You said that they allow the
Speak so that you can.
yeah, you said that they allow things like cytoplasmic to mix. So I wanted to find out, according to cell physiology, there are certain specialized cells. In cytoplasm generalized, whereby cytoplasm for the digestive system, the cell there, can mix with the cytoplasm for, let's say, the respiratory system.
Okay, I only need 2 minutes to answer the question. Any other questions? Do you have any other questions? Now, I'm going to answer your questions. I've gotten them. But I want to answer them at the same time because we're on a 5 minutes break, remember? Any other question?
There's a hand.
Where?
Yeah.
Oh. I hope I wasn't too fast. If I was too fast,
Since you say you were supposed to differentiate the three that you talked about earlier, the ones that connect the cells. Is it that all the three are connected? Let's say, for instance, this cell, are they all there at the same time, or they interchange, or it depends on the two cells that are
kind of related to her question, right?
Yes.
So, any other question? I'm going to answer them in 3 minutes, and we'll start the lecture at 12. So, her question is more related to her question. So, as we'll be answering her question, we'll also be answering her question.
So my question is junction. When you look at the side junction, and then there's the other one, the adherent junction. So in the notes I've observed, the one who's talking about the tight junction. Does that mean that the adherent is under the tight junction as well? Or there's a
Okay, again, I've answered your question. I'm taking, this is the last minute of taking questions. Any other questions?
Since you have talked a lot about what about if they ask you to talk about the same are you supposed to like 2 pages or
In my third year, I wrote a paragraph of I wrote 1 page and half paragraph, and I got the 0. My other roommate, I remember
Janet.
coming to cry because our seniors were very, very bad, like terrible. My other roommate wrote about 2 pages and got 0.5 out of 20. What am I driving to? You need to understand that physiology is interested in function and not structure. But for us to explain function, we have to go a little bit to structure. So in most cases, you find I write 1 whole paragraph describing structure, and then I have nothing to talk about function. Have you understood the difference? So, to answer your question, it depends on the question that you're being asked. But as you answer your question, try not to go to lean more on structure, lean more on the function. Then we all know Drew, right? The guy who had like 20 points. You never heard, the guy who graduated with a distinction. Oh, come on, you don't know him.
We know him.
You know him. That guy wrote half a page and had 19 out of it.
That one is such a laugh.
He's going to come and teach you. I think we should do something. After this, after cell physiology, I'm sure it's Drew should be coming. I should be going to pay this. So you're going to interact with one of the best students. I respect it, it's very good. But you asked me to answer your question. Number one, in physiology, related to function, not structure. We can talk a bit of structure, but talk in terms of function. Okay. Then, to answer all your questions now combined, I have 1 minute to do that. I talked about the cytoskeleton. I talked about the cell adhesion molecules. I talked about the gap junctions. In my starting, maybe I was too fast. What I said was, all these molecules, all these three structures are being formed by three basic components of the cytoskeleton, which were the microtubules, the microfilaments, and the intermediate filaments. So your cytoskeleton is like an umbrella of the cell adhesion molecules, your adhesion or your all those junctions we talked about, and the gap junctions. Why? Because your cytoskeleton is what is comprising of those filaments. Cell adhesion molecules are made up of microtubules. They are made up of microfilaments. All these are components of your cytoskeleton, correct? So your cytoskeleton is any of these structures that are going to hold the cell together. Now these structures specialize. Some are going to form channels between two cells, and we're going to call those gap junctions. They are holding cells together, tightly together, but at the same time, they are allowing cytoplasm to pass through. We call those gap junctions. They are those that are found on the apical surface, okay, that are holding your proteins together. We call those our tight junctions. Then we had a continuation. So on the apical side, we say they are preventing proteins to move unnecessarily in the, what plane is this again?
The vertical.
Vertical, isn't it? And then you had some adhesion molecules that were preventing proteins to move in your horizontal. And I mentioned to say you have your zonular, the desmosomes, that are basically holding cells together in the lateral direction. But all these are still components of your cytoskeleton. Why? Because your cytoskeleton is holding cells together. Okay? Then I talked about cell adhesion molecules. They are still falling under your cytoskeleton because, first of all, they are made up of microtubules, microfilaments, intermediate filaments, and they are providing stability to the cell. Only that this time, your cell adhesion molecules are joining cell to cell. They are on the apical surface, on the outer surface of your cell membrane, and they are holding two cells together. And therefore, we're now going to call them cell adhesion molecules. They are holding one cell to another, apically, outside. So your cytoskeleton is both inside the cell and outside the cell. Then we now start dividing based on what they are, where they are found, and on their function. Okay, does that try to answer your question? For his question, what was your question again? Oh, how cytoplasm moves. Remember, he said that cells are grouped together to perform similar function. So you're not going to have the cell cytoplasm of heart cells and respiratory cells. Why? Because first of all, these are different organs. They have similar shape, they have similar organelles. In my introduction, I mentioned that based on the type of cell, they are going to have different concentration of organelles in those cells. Remember that statement? So they are different. Secondly, cytoplasm, remember, we say it's made up of what? 80% of water. The only difference is the concentration of ions that are present. which is dependent on the function. So the concentration of the cytoplasm is different from one organelle to another organelle based on the function. Does that answer your question? And they don't mix, no. Is there a question I haven't answered? Okay, so I hope I've tried to clarify cytoskeleton. Very, very important. Now, we have to quickly rush through organelles. I don't mind if I don't finish this because I know that at least you have an idea of what it is and you can go through it at your own time. We have a nucleus, okay? So the nucleus, we all know, okay, maybe some of you don't know, that the nucleus contains DNA or your genetic material in the form of, genetic material in the form of DNA and RNA, isn't it? And then, of course, you have it being transmitted to the cell through your mRNA, okay? So it contains the nucleus except matured red blood cell. Underline the word matured. So before a red blood cell matures, it has a nucleus. So if these things are they look very simple, but when you come to the hospital, you'll be forgetting them. When you get a malaria slide, not a malaria slide, a blood slide, okay? And then we find that there are red blood cells with a nucleus. Then we start thinking of things like aplastic anemia, okay? So these have clinical implications. So you don't expect to find a nucleus in a mature red blood cell. So all cells have a nucleus except for matured red blood cells. In terms of size, we're saying that it's the largest. Of importance, it is bound by two membranes, okay? So I mentioned earlier to say all the organelles have a membrane that binds them. You have two organelles in the cell which have a double-layered membrane around them, which is your nucleus and your mitochondria, okay? We call that a nuclear envelope. The nucleus contains genetic material, and that material is DNA, isn't it? Of course, you have your enzymes and your proteins that are important, your enzyme gyrase, those funny enzymes. People are doing that last year, did? Yeah, so those enzymes, you find them together with your RNA and your mRNA. It contains a nuclear lamina, a nucleolus. So the nucleolus is more like that's your, okay, let's start with the nuclear envelope. The nuclear envelope there, we say that there are two membranes that surround the nucleus. In terms of diameter, again, this is an electron microscope. We might never see this. I've never seen a nucleus, like when you narrow it down to see the membrane. Okay? It contains in diameter. This nuclear envelope of importance is that it joins or it connects to the endoplasmic reticulum. And we're going to see the reason why when we start looking at what? The endoplasmic reticulum. In terms of the nuclear lamina, these are just proteins that are attached to chromatin in the inner membrane of the nuclear envelope. So you have two envelopes, meaning that one is on the outside and one is on the inside. When we say outside, we mean in the cytoplasm, isn't it? And then when we say inside, we mean the area which has your DNA or your genetic material. Okay. These lamins are attached to chromatin in the inner membrane of the nuclear envelope and participate in breakdown and formation of the envelope during the cell cycle. So the nuclear envelope is regulated during the cell cycle. We're going to look at the cell cycle in another lecture and how it is regulated. One of the ways is these important proteins, okay? They're important in breakdown of certain nuclear envelopes. And we'll particularly see the significance of that in later discussions. In terms of the nucleolus, it's responsible for ribosomal RNA synthesis. And we know that the ribosome is important for what? Protein synthesis, isn't it? Okay. Of course, it contains two zones. You have the granular zone in the peripheral, and then, is it three zones? Three zones, sorry. You have your granular zones. What differentiates rough and smooth endoplasmic reticulum? The presence of ribosomes, isn't it? Okay. So you have that granular zone, basically where you're having the ribosome assembly. You have the fibrillar zone, which is located. Do I have a picture of this? Unfortunately, I didn't put a picture. Okay, I'll give you these slides, but the ones I'll try and send other ones, maybe with the picture to illustrate that. Okay. And then you have the fibrillar center zone that contains DNA that is not being transcribed. Okay. In terms of chromatin, again, I take you back to your first year. You see why I say that we do this at the end? Because we all remember what chromatin looks like, correct? Basically, it's a complex of DNA. So that DNA strand, you have one DNA strand and another DNA strand. So that complex, we call it a chromatin. It has the DNA strands. It also has histone proteins and non-histone proteins. We all know that the DNA is a double-stranded strand helical molecule. Sorry about that. In terms of the conformations, don't worry about yourself with those conformations. But you need to remember that it has histone proteins. We know that histone proteins are what? Are positively charged. Okay. And they are enriched with lysine and arginine residues. Again, these are just nitties. Lysine, we all know what do we call those things again? They are amino acid sequences, isn't it? Like, have we done our biochemistry already? So we understand what lysine and arginine is? Okay, that's okay with me. Okay. Of course, at the center you have your what? Your nucleosomes, which are basically repeating units of the chromatin, or repeating units of the same genetic material. Non-histone proteins, I'll take you back to your first year again. You have those enzymes that are responsible for replication, transcription, and DNA repair. Okay. We move on quickly to the endoplasmic reticulum. We mentioned earlier that the endoplasmic reticulum is directly connected to your nucleus, and based on the presence of ribosomes, you have two types. You have the rough endoplasmic reticulum, you have your smooth endoplasmic reticulum. Please remember that you have some free ribosomes that are present in your cytoplasm, and those are important for protein synthesis. So based on the presence of ribosomes, you have your rough or your granular endoplasmic reticulum, and then you have your smooth or your granular endoplasmic reticulum. Is it 4:15 already? 4:13. 3 minutes. Two, huh? Okay, a granular endoplasmic. So let's make a decision. Should we end here and we come and finish up organelles in the next lecture, or we finish them up?
No. Next lecture we need to finish. Another thing will come.
Oh, you are going to go and read.
We can read. These we can read.
Okay, now that's a few slides. I can come and talk about them in the next lecture. I think we've been talking for a very long time, so let's not
Start.
people too much. Yes. Oh, sorry. As I answer your question, so please, as you go home, I'm sending the slides to your classroom just now. You can go through these organelles, but I'll come and quickly peruse them when we meet next time. I think we've been talking for a long time, unless there's a process that we finish.
Okay, my question was ribosomes. concerning the ribosomes, those which are attached to the graph and the ones which are pre I mean those which are pre the cytoplasm. So what is the greatest advantage, or should I say, what special function to those which are attached to the ER before? What is the greatest function?
Class is dismissed, guys. We're free to go. I'm just taking questions from those who have questions. I understand you have another class, correct? What time is it?
Another 14.
What?
14 to 16. He was one for
So you are not going anywhere. Why can't you? Okay, fine. We are free to go, please. We are free to go. The class is done.
Just please ensure, like, as soon as I share it, but that's the